Welcome back to the deep dive.
Hey there.
So today we're not just skimming the surface, we're going deep.
All the way to the bottom on this one, yeah. We're tackling a biological mystery that has, frustrated scientists for decades.
We are talking about tauopathies.
It really sounds like a sci-fi villain, doesn't it?
It does, the tauopathy.
But it's actually, a whole category of neurodegenerative diseases. You've got Alzheimer's, which everyone knows.
Of course.
But then there's CTE, chronic traumatic encephalopathy, that affects athletes.
Right.
You have corticobasal degeneration, CBD, Pick's disease.
And here's the hook, right? The thing that always gets me. We know that all of these diseases, fundamentally, are caused by the exact same protein, tau. It goes rogue.
It clumps up, tangles, kills brain cells.
But if it's the same protein, why does Alzheimer's look so different from CTE? Why does Pick's disease hit your personality while CBD messes with your movement?
Exactly. Why does the same ingredient cook up so many different recipes for disaster? For a long time, we've just been looking at it with a magnifying glass. We just see clumps.
But today we're switching to an electron microscope.
You could say that.
We've got a source today that claims to have finally cracked this code. We're looking at a groundbreaking paper. It was published in Cell in 2026 by Kumar and his colleagues.
And they didn't just look at a few mice in a cage.
No. They analyzed 203 human brains.
That's a massive cohort for this kind of work. Usually these studies have 10 brains, maybe 20. Tauopathy is a data goldmine.
And their mission was just incredibly ambitious. They wanted to create a molecular map of tau for every single one of these diseases. And they used this tech called FLEXITau.
Right. And to really get why this matters, you have to understand that our standard methods, they just ask, is tau there? It's a head count.
Yes or no?
A FLEXITau is totally different. It's a type of mass spectrometry, but it asks what is written on the tau. It literally reads the chemical graffiti on the protein.
I love that image. Chemical graffiti. Okay, so we're going to go through this paper, sentence by sentence, detail by detail. No skipping the hard stuff today.
Let's do it.
Here's our roadmap. First, the hierarchy of clumpiness, who has the most tau. Second, the shapeshifters, how the protein is spliced. Third, the chemical graffiti we just mentioned, modifications, shredding. And finally, how a machine learning algorithm used all this to diagnose patients better than a doctor could.
It's a lot to unpack, but, the details are where the magic is.
Okay, let's start with part one, the hierarchy of pathology, or what I'm calling the trash pile.
It's an apt analogy. The researchers took these 203 subjects, AD, familial AD, CTE, CBD, Pick's, PSP, and controls, and they measured the absolute abundance of an insoluble tau.
So basically, how much of this protein has turned into hard, undissolvable junk in the brain?
Precisely.
And the differences were staggering. I was looking at the numbers. Let's just walk through the ranking. Who is the king of the trash pile?
Oh, without a doubt, it is familial Alzheimer's disease, or FAD.
The genetic cases.
The genetic ones, exactly. And they found, get this, about 3,450 femtomoles per milligram of tau.
3,450. Okay, and for everyone listening, femtomol sounds tiny, but biologically that is a mountain. How does it compare to, say, regular Alzheimer's?
Regular Alzheimer's is still incredibly high, about 12,220.
Okay.
To put that in perspective, that is roughly 81 times more aggregated tau than you find in a healthy control brain. It is a massive physical burden.
81 times.
Imagine your house filled floor to ceiling with trash. That's the Alzheimer's brain.
Wow. Okay, so AD is the heavyweight champ of clumps. Who's in the middle tier?
In the middle, you have CTE. That's the concussion-related one at about 190, and CBD at roughly 150.
So what, 10 to 12 times more than controls?
About that, yeah. Significant, but just nowhere near the sheer volume you see in Alzheimer's.
And the lightweights, who had the least?
And this was the surprise. PSP progressive supranuclear palsy and Pick's disease. They were sitting at around 30 femtomoles per milligram.
Only 30. That's barely anything.
It's only about 2 times the baseline of a healthy control.
That is wild. PSP is a devastating disease. It kills people quickly, and yet physically there's way less junk in the brain than in Alzheimer's.
Correct. And that is a key insight we're absolutely going to come back to. We used to think more plaque equals more disease.
But this suggests that might be way too simple.
Or just wrong. High abundance seems to correlate with something else, extensive modification.
Yeah.
The AD tau is chemically a mess. The PSP tau is relatively clean.
Okay, so that's the amount. Now let's talk about the type of tau. This brings us to concept 2, isoforms, the splicing blueprint.
Right. So quick biology 101. Your DNA makes RNA, and that RNA gets spliced, kind of like editing a film reel to make the final protein.
Right.
For tau, the key scenes being edited are called exon 2, exon 3, and exon 10.
Let's focus on exon 10. That's the famous one. This determines if the tau has 3 repeats or 4 repeats in its binding domain. The 3R versus 4R ratio.
And this is the first major filter for sorting these diseases. CBD and PSP are what we call 4R dominant, meaning their ratio of 3R to 4R is very low, like 0.1 to 0.3. They almost always include that exon 10, making them 4R.
Okay, so CBD and PSP are team 4R.
Exactly. On the complete flip side, you have Pick's disease, that's team 3R. Its ratio is around 1.6. It mostly excludes exon 10.
And what about the big ones, Alzheimer's and CTE?
They're the diplomats, they're mixed. Their ratio sits right around 1, so they have a pretty even balance of both 3R and 4R tau.
There was one more splicing detail that stood out to me from the paper. The N-terminal splicing with exons 2 and 3.
Yes, and this is crucial. You can have 0N where both are missing, 1N or one's there, or 2N with both present. And the study found that in all of these tauopathies,
All of them?
all of them, the insoluble tau, the bad stuff, was dominated by the 0N isoform.
So the shortest version of the protein's head, where both exon 2 and 3 are chopped out of the RNA, that's the stickiest one.
It really seems to be. The 0N isoform is the one most prone to falling into that trash pile across every single one of these diseases.
Okay, we are about 5 minutes in. Let's do a quick recap of part 1 just to make sure we've got this locked down.
Let's do it.
Bullet 1, we're looking at a huge study. 203 brains, 6 distinct diseases plus controls.
Bullet 2, abundance is a spectrum. Familial AD and AD have massive amounts of tau-like 81 times the baseline. PSP and PICs, surprisingly little, only about 2 times.
Bullet 3, the isoforms, they separate the tribes. CBD and PSP are 4R diseases, PICs is a 3R disease, and AD and CTE are mixed.
And bullet 4, the 0N isoform, the short one, is the universal troublemaker. It dominates the aggregates in every single disease we've talked about.
Perfect. Okay, now let's get microscopic. We're moving to part 2, the chemical graffiti and the shredder.
This is where FLEXITau really shines. We're talking about post-translational modifications, or PTMs.
Little chemical tags added to the protein after it's made?
Exactly. The paper calls the enzymes writers and erasers. Writers add the graffiti, erasers scrub it off.
And how many of these graffiti tags did they map?
145. 145 different PTM sites. It's an incredibly complex landscape.
Okay, let's break down the types of graffiti. First up, phosphorylation. That's the classic one everyone studies, isn't it?
It is. It's the most common. But they found this really neat distinction between universal tags and specific tags. For example, phosphorylation at a site called serine 202, PS202. It was everywhere. Every disease had it. Even the healthy controls had a little bit.
So PS202 is like the hello world of tau modification. Doesn't tell you much.
Exactly. It just tells you tau is here, not what disease is this. But then you look at, say, threonine 231 or PD231. This one was present in all the disease subjects, AD, PSP, CTE, all of them. But it's completely absent in the healthy controls.
Ah, so PT231 is a disease flag. If you see it, something's wrong, but you still don't know what's wrong.
You've got it. It's a general alarm bell.
Okay, so how do we start telling them apart?
You have to look at the other types of graffiti, acetylation and ubiquitination. And these are really interesting chemically because they change the protein's electrical charge.
Okay, physics time. The paper talks about the microtubule binding domain, the MBD. Normally, this region has a positive charge, right?
Correct. And since positive charges repel each other, like 2 north ends of a magnet pushing apart, healthy tau molecules don't want to clump up.
They push away from each other.
But in Alzheimer's,
Uh-oh.
In Alzheimer's and CTE, the study found massive amounts of acetylation and ubiquitination right in this region.
Right.
And these chemicals, they neutralize that positive charge.
And when you neutralize the charge,
You lose the repulsion, the magnets stop pushing,
Right.
the proteins can just collapse onto each other and start stacking. It stabilizes the whole aggregate.
so that's the glue.
That is the glue. The study points specifically to acetylation at lysine 281 and ubiquitination at lysine 353.
And CBD had its own specific fingerprint here too, right?
It did. CBD had a very specific ubiquitination at that same lysine 353 site, but the pattern looked distinct from AD. That's how you start to tell them apart chemically.
They also mentioned citrullination.
All right, at arginine 406. This was highly frequent in AD and familial AD, but pretty rare in the others. Just another clue for the detectives.
Okay, that's the graffiti. Now let's talk about the shredder. Proteolytic cleavage. This is just enzymes chopping the protein backbone into pieces.
They mapped 195 cleavage sites. So the protein is just getting hacked up. But the pattern of the hacking is totally different between diseases.
A paper talks about hotspots, areas where the molecular scissors cut over and over again, like amino acids 355 to 361.
Exactly. Now, I want you to picture Alzheimer's tau. The study describes it as extensively cleaved.
Meaning?
The N-terminus, the head of the protein, is chopped off, gone. The C-terminus, the tail, is also chopped off. You are left with just the core, that microtubule binding domain.
So AD-tau is like a shredded document where only the most important paragraph in the middle is left.
That's a perfect analogy. And that core is extremely stable. Because the floppy ends are gone, it stacks perfectly. Now contrast that with PSP.
Which they describe as minimal cleavage.
Right. In PSP, the protein is mostly full length. It's not shredded. Maybe a little crumpled. But the N-terminus is still there. The head is still attached.
And does that explain why there's less of it in the trash pile?
It seems to. The N-terminus is floppy and bulky. If it's still attached, it acts like a bumper on a bumper car. It physically prevents the proteins from stacking too tightly.
But if you cut it off, like in AD...
They stack like bricks.
So to visualize, AD-tau is a stack of bricks shredded cores. PSP-tau is a pile of bumper cars full-length proteins.
That's the visual that makes it stick.
Okay, let's hit recap number 2.
Ready. Bullet 1, PTMs. They found 145 sites. Phosphorylation is everywhere, but it's the acetylation and ubiquitination that really drives stability in AD and CTE by neutralizing that charge.
Bullet 2. Universal versus specific. PS202 is in everyone, but acetylation at K281 is more specific to those heavy clumping diseases.
Bullet 3. The shredder. AD and CTE tau is heavily cut up, losing its head and its tail. PSP tau is mostly left intact.
Bullet 4. The mechanism. So cutting off the floppy ends and neutralizing the charged middle, that's the recipe for a massive, stable plaque.
Precisely.
Okay, on to part 3, the proteoform fingerprint and machine learning.
Right, this is where the paper gets very high-tech. They used FLEXITau to generate these beautiful heat maps.
And why wasn't binary data enough? Why couldn't they just say, yes, it's modified, or no, it isn't?
Because biology isn't binary, it's continuous. You need to know the stoichiometry, basically, the extent of the modification.
Okay.
Is 10% of the protein modified at this site, or is 90%? FLEXITau can measure that.
And they visualize this with colors. Blue meant low modification, mostly normal. Red meant high modification. It's been chemically changed or cut.
And when they fed this data into a computer, without telling the computer which patient had which disease, by the way.
Unsupervised,
the computer clustered them perfectly.
Let's talk about those clusters. What was cluster F6?
F6 was the Alzheimer's cluster, and it was red hot. I mean, 90% plus modification across almost the entire protein, just a complete chemical mess.
And cluster F3?
That was PSP, and it was fascinating to look at. The N-terminus was bright blue, unmodified, intact, just like we said. But then the C-terminus had these very specific red bands of modification.
A totally different visual pattern.
Completely different.
So they took these patterns and they trained a machine learning model on them, a random forest algorithm, how to do.
It was impressive. It achieved an AUC, that's area under the curve, of about 0.86.
Which means, in plain English,
it means it could distinguish these diseases with very high accuracy based only on the chemistry of the tau protein.
Wow. What were the key features the robot was using to tell them apart?
Well, for Alzheimer's, the computer looked for that classic profile. High abundance plus cleavage plus massive PTMs.
The shredded red-hot mess.
Exactly. For CTE, it found a really subtle difference. The proline-rich region, or PRR, had specific spots that were unmodified, whereas in AD, they were modified.
That's interesting. So CTE has a clean spot that AD doesn't have. What about Pick's disease?
The algorithm honed in on these unique modification patterns at amino acids 195 to 209, and then again at 384 to 395. Specific zones that only light up in Pick's.
And CBD?
It used that 4R dominance we talked about, plus that heavy ubiquitination right in the binding domain.
So we have a digital pathologist now that can read molecular graffiti.
We do, and it's seeing things human eyes would almost certainly miss. It proves that each disease has a specific proteoform fingerprint.
Time for a recap. Number 3.
Bullet 1. FLEXITau gives us the extent of modification from 0 to 100%, which is critical for telling these diseases apart.
Bullet 2. Machine learning actually works. The models can diagnose the specific type of dementia just by looking at the protein's chemistry.
Bullet 3. The AD profile is shredded and red, high abundance, heavily cut, heavily modified.
Bullet 4. The PSP profile is blue and intact. Low abundance, the N-terminus is fine, but the tail gets modified.
You're getting good at this.
I'm learning from the best. All right, part 4. the synthesis, the so what. Why does any of this matter for the person listening who might be worried about their brain health?
I think the biggest takeaway is about diagnostics and drugs. Right now, in pathology, we use an antibody called AT8 to stain brains.
Which binds to PS202 and PT205.
Right. And as we just learned, PS202 is in everyone. It's a blunt instrument. It tells you there is tau, but it doesn't tell you this is CBD or this is CTE.
So this paper argues we need biomarkers for precision medicine.
Exactly. And think about drug development. If you're developing a drug for Alzheimer's, you might need an eraser enzyme that targets the acetylation in that microtubule binding domain to stop the stacking.
But if you give that same drug to a PSP patient,
It probably wouldn't work. It might do nothing at all because PSP tau isn't really acetylated in that region.
or say you try to stop the shredding enzymes, the proteases, that might help in Alzheimer's where the protein is shredded.
But in PSP, the protein isn't being shredded anyway, so blocking the shredder is completely useless.
So the one ring to rule them all, one-size-fits-all approach to curing dementia is dead.
I think this paper drives a stake right through its heart. We need anti-A-tau drugs and anti-PSP tau drugs. They're just different molecular enemies.
The paper introduces this idea of the proteoform theory.
It does. It's the idea that the specific shape and chemistry, the proteoform, is what dictates the final structure of the fibril. We've known from other studies, from cryo-EM, that the fibrils look physically different. This paper explains the chemical reason why they look different.
It connects the chemistry to the structure. The chemistry guides the folding.
You got it. That's the link.
Okay, we're nearing the end. I want to leave our listeners with the provocative thought from the outro. And this is a bit of a paradox the authors bring up.
It's probably the most mind-bending part of the whole study. We established that PSP progresses faster than Alzheimer's. It's a more aggressive, more rapid disease.
Right. But chemically, PSP tau is less modified, it's less cleaved, and there is way, way less of it in that trash pile.
Exactly. So in AD, you have these massive, stable brick wall plaques. In PSP, you have these small, unstable, floating oligomeric bits.
So here's the hypothesis. Maybe the plaque in Alzheimer's is actually good.
Good might be a stretch, but maybe protective.
Protective.
It's possible that the brain is actively shredding the tau and gluing it together into these giant inert clumps to lock it away, like putting nuclear waste into a concrete sarcophagus.
And in PSP, for some reason, that mechanism fails. The tau stays fresh, unstable, and oligomeric. And because it isn't locked down, it can spread from cell to cell much, much faster.
The unstable bumper car tau of PSP might be more toxic precisely because it's free to roam and infect other neurons. The shredded documents of AD are stuck in the filing cabinet.
That completely flips the script on therapy. We have spent billions of dollars in decades trying to break up plaques.
Right, but if you break up the plaque, are you just releasing the toxic genie from the bottle?
And that is the question we want to ask you, the listener. Based on this deep dive, if rapid fibril formation is actually the brain protecting itself by locking away toxic tau, are we making a huge mistake by trying to dissolve them? Should we instead be trying to seal them faster?
It's a question that could determine the entire future of dementia treatment. It's that big.
And one more call to action. We talked about heat maps today, blue and red. In the future, your medical record might not just say dementia. It might have a FLEXITau heat map of your specific protein signature.
Personalized molecular maps. That's the future. That's where this is headed.
Fascinating stuff. As always, thank you for listening.
Keep thinking critically.
And keep diving deep. We'll catch you next time.